We all rely on medicines, to get better. But
Yeah.
have you ever really stopped to think about how that tiny pill actually triggers such specific changes inside your body? It's this incredibly complex dance happening right down at the molecular level.
It really is.
Yeah.
And for the longest time, getting a clear view of that dance, actually seeing how drugs hook up with their targets, well, that was a huge scientific challenge, a real bottleneck.
Right.
But that's exactly what we're diving into today. We're looking at some really groundbreaking research published in Nature over the last few years, say 2020 to 2025. And it's all thanks to this revolutionary technique called cryo-electron microscopy, or cryo-EM for short.
Cryo-EM. Okay, sounds pretty high tech. So break it down for us. What does it let scientists actually do?
Well, imagine you could take a tiny biological machine, like a protein that a drug is supposed to target, and flash freeze it, like incredibly fast. So fast that it's just frozen solid in its natural shape, mid-action almost. Then you use an electron microscope, bombard it with electrons, and use really powerful detectors and clever software to build a 3D picture of that molecule.
So it's like taking this super detailed 3D snapshot, freezing these tiny players right in the middle of doing their job. What were scientists doing before this? Why is cryo-EM such a leap forward?
Yeah, the main method before was something called X-ray crystallography. And that meant scientists had to kind of coax these molecules, usually proteins, into forming these really stable crystals first.
Ah, I remember hearing about that. It sounds tricky.
It can be incredibly difficult, especially for molecules that are flexible or, embedded in the cell membrane, the cell's outer layer. And a lot of really important drug targets fall into that category. They were just hard to study.
So cryo-EM gets around that.
Exactly. It skips that whole crystallization step. So we can study a much wider range of molecules, and crucially, in a state that's much closer to how they actually exist and function inside our bodies. That's particularly important for those drug targets we couldn't really see properly before.
Right, okay. So our mission today, then, is to dig into these Nature papers and really see how this amazing visual detail from cryo-EM is letting us, literally see how drugs do their job. We want to understand the architecture, the structure of these targets, how the drugs actually latch onto them, what happens next to cause and effect, and ultimately, maybe the structural reasons why some drugs work really well and others don't. Is that about right?
That's precisely it. We're focusing on those fundamental molecular interactions, the ones that really determine if a drug is going to be effective. So where should we start? Maybe ion channels?
Ion channels, yeah, I remember learning about those. They're like crucial gateways in our cells, right? Involved in nerve signals, muscles, loads of things. And they're a major class of drug targets, aren't they?
They absolutely are. Really vital drug targets. But again, because they're often flexible and stuck in membranes, they were very difficult to study structurally using the older methods. Cryo-EM has been a total game changer here.
Okay.
Let's take the glycine receptor, or GLIR, as a good example.
GLIR. Okay, what have we learned about that one thanks to cryo-EM?
Well, these cryo-EM studies have let us visualize GLIR in these really short-lived intermediate states, like the exact moment just before the channel fully opens.
Wow.
And what's really insightful is seeing how partial agonists, those are drugs that only produce a partial effect, not the full response.
Right, they don't quite work fully.
Exactly. We can see that they tend to get the receptor stuck in these states where the agonist is bound, but the channel is still closed. So cryo-EM basically shows us the structural reason why these drugs aren't as effective.
So it's like they fit the lock okay, but they just can't quite turn the key all the way to open the door.
That's a great way to put it, yes. And this insight, this structural detail, is key for designing future drugs, modulators that might have better efficacy because we know what conformation to avoid or target.
Makes sense.
And beyond that, we've also visualized GLIR in its fully open state, its completely closed state, even its desensitized state when it stops responding. And we can see these states with different molecules bound, like glycine itself, or taurine, or GABA.
So you get the whole picture of its activity cycle.
Pretty much. It gives us this incredibly detailed view of how the channel changes shape as it does its job. And that level of detail is just invaluable if you want to design drugs that can selectively fine-tune GLIR activity, precision targeting.
Yeah, precision is definitely the name of the game. Okay, what about those voltage-gated sodium channels, the NaV channels? I know they're critical for how we feel pain.
Absolutely. NaV 1.8 is a specific one that's really important in pain sensation. So it's a major target for painkillers or analgesics.
Right.
And cryo-EM studies have shown us precisely how different drugs Things like suzetrigine A887A26, LTGO33, how they inhibit NaV1.8. And importantly, they do it in a state-dependent manner.
State-dependent, what does that mean exactly?
It means the drug works differently depending on the electrical state of the nerve cell, whether it's resting or active. And interestingly, Cryo-EM has actually revealed the structural basis for something called reverse use dependence that we see with some inhibitors.
Reverse use dependence sounds backward.
It kind of is. It means when the voltage sensors in the channel get depolarized, basically, when the nerve cell is becoming more active, firing more signals, some drugs actually bind less well, their affinity goes down.
Huh. So when you most want the painkiller to work, it works less effectively.
In some cases, yes. And understanding the structural reason why this happens, which Cryo-EM has shown us for NaV1.8, is now helping researchers design new pain meds that hopefully won't have this limitation.
Wow, okay. Seeing that 3D structure really explains the why behind that counterintuitive effect. That must be incredibly useful for designing better pain relief.
Hugely useful. Then there's another interesting one, TRPV1, the vanilloid receptor. You might recognize it as the thing that responds to capsaicin.
Oh, yeah, chili peppers. So Cryo-EM helped figure out why my mouth feels like it's on fire.
Huh. In a way, yes. TRPV1 is involved in pain and inflammation pathways. Cryo-EM let us visualize the very specific shape changes TRPV1 goes through as it moves from being closed to being open when capsaicin or similar molecules bind.
You can see the whole process.
We can see the trajectory, like the step-by-step path the channel takes as it opens up at the molecular level. And understanding that whole activation pathway is really crucial if you want to develop drugs that target TRPV1 effectively for pain or inflammation.
So looking across these different ion channels, GIR, NaV1.8, TRPV1, the big story seems to be Cryo-EM giving us this unprecedented clarity. We can see the different shapes they take, exactly how drugs interact. Feels like moving from guesswork to actual design.
That's a very good summary. It's moving drug development from a more empirical, trial and error approach towards a much more rational, structure-guided one. If you look at the key findings just from 2020 to 2025, for GIR, it's those intermediate states, why partial agonists are partial. For NaV1.8, it's that state dependence mechanism. For TRPV1, it's the opening trajectory. All fundamental structural knowledge for rational drug design.
Okay, so ion channels, definitely a big impact area. What about GPCRs? That's another huge family of drug targets, right? Involved in just about everything.
Oh, absolutely. G protein-coupled receptors. They're the single largest family of drug targets we have, involved in an enormous range of body functions, and targeted by a huge percentage of the drugs currently on the market.
But historically tricky to study, I imagine, like the ion channels.
Exactly the same issues, really. They're membrane proteins, often quite flexible, unstable. Getting them to form those nice crystals for X-ray methods was, well, notoriously difficult for many of them.
Another perfect challenge for Cryo-EM, then. Did it deliver?
It really did. Cryo-EM has genuinely revolutionized GPCR research. We can now get these 3D structures in all sorts of functional states, bound to different signaling molecules, different drugs, activating protein. It's opened up whole new ways to understand how they work and how we can target them.
Can you give us a specific example, a recent breakthrough maybe?
Yeah, sure. A really interesting one is the study of the arginine vasopressin V2 receptor, V2R.
Yeah.
Cryo-EM allowed researchers to solve its structure when it's bound to a really important signaling partner called V-arrestin-1. Now, GPCRs signal through G proteins, but V-arrestins are also crucial. They regulate the receptor, turn it off, but also start their own signaling cascades.
Right, arrestin-CRs are key players too.
They are. And what the Cryo-EM structure showed was actually quite surprising. V-arrestin binds to V2R in an atypical position compared to how it binds to other GPCRs we've looked at.
Oh, so it's not always the same interaction.
Apparently not. It suggests there's actually significant structural diversity in how GPCRs and arrestins hook up. Our previous models might have been a bit too simple. This could open up completely new ways to fine-tune GPCR activity.
So maybe targeting that specific, unique interaction?
Potentially, yes. The Cryo-EM data showed exactly which parts of the V2R are involved, all the intracellular loops, specific phosphorylated bits on its tail interacting with a particular part of arrestin. Getting that level of molecular and structural detail is just essential for understanding how V2R signaling is controlled.
And how does knowing this very specific V2R arrestin structure help develop better drugs?
Well, having these high-res GPCR structures in various states lets us design new ligands, the molecules that bind with much, much greater precision. We can literally see where a drug needs to dock to get the therapeutic effect we want.
Leading to better drugs, hopefully.
That's the goal. Better potency, better selectivity, hitting just the target you want and not others, and ultimately better effectiveness. It's very interdisciplinary now, combining cryo-EM with other methods for diagnostics and therapies. And for V2R specifically, understanding this unusual Y-arrestin binding, thanks to cryo-EM, gives us new ideas for regulating it.
So cryo-EM isn't just giving us, nice pictures. It's fundamentally changing how we approach drug development for this massive class of receptors, accelerating things.
Exactly. It provides the essential structural blueprints we need for rational design. It's speeding things up significantly.
Okay, now another really big challenge in medicine is drug resistance. We hear about antibiotic resistance all the time. Cancer's evolving to resist treatments. It's a huge threat. How is cryo-EM helping us understand and maybe fight back against resistance?
Yeah, drug resistance is a critical issue, definitely growing. And cryo-EM is proving to be a really powerful tool for figuring out the structural changes that actually cause that resistance. Let's take antibiotic resistance as a really urgent example.
Definitely a major global health concern.
So one key way bacteria resist antibiotics is using things called efflux pumps. These are basically proteins in the bacterial membrane that actively pump the antibiotic molecules back out of the cell.
Ah, so they just kick the drug out before it can work.
Pretty much. They stop it reaching its target inside the bacteria. And cryo-EM has let us determine the 3D structures of these efflux pumps, while they're actually bound to different antibiotic molecules.
So you can see the pump grabbing the antibiotic.
We can see the snapshot of that interaction, yes. Where exactly the antibiotic binds inside the pump, which specific amino acid residues of the pump protein are involved in holding it, the whole setup.
Okay.
And this detailed view helps us understand how mutations, small changes in the pump protein sequence, can alter its structure. Maybe the antibiotic doesn't bind as well anymore, or maybe the pump gets even better at throwing it out. That's how resistance develops at the molecular level.
That's incredible insight. So if you can see how the pump works and where the antibiotic binds, maybe you can design what? New drugs the pump doesn't recognize. Or maybe drugs to block the pump itself.
Both, exactly. Block the pump, and maybe old antibiotics become effective again. Or design new antibiotics that evade the pump. And this isn't just for antibiotics. Cryo-EM is also being used to study resistance in cancer therapy.
How does that work in cancer?
Well, by looking at the structure of the drug target in the cancer cell, we can try to identify areas or interfaces that are less likely to mutate. If you can design a drug that binds across a larger, more stable area of the target,
then it's harder for a single mutation to just knock it off and cause resistance.
Precisely. Visualizing these broader drug target interfaces with cryo-EM helps design drugs with a more robust grip, making resistance less likely to pop up. So the bottom line is cryo-EM is giving us the structural intelligence we need to develop new ways to tackle resistance.
So we've gone from seeing the targets to how drugs bind, and now even how targets change to resist the drugs. It really feels like we're building this incredibly detailed map of molecular interactions in disease and treatment, which must be invaluable for structure-based drug design, which you mentioned earlier.
Absolutely. Structure-based drug design is, fundamentally, using that 3D structure of a biological target, like a protein involved in disease, to guide the whole drug discovery process. And cryo-EM is now providing those critical high-resolution starting structures.
Can you walk us through how that process generally works using cryo-EM?
Sure. It often starts by identifying specific pockets or binding sites on the target protein using the detailed map from the cryo-EM structure.
Okay, finding the docking spots.
Exactly. Then researchers use powerful computers to either design new molecules or screen huge virtual libraries of existing ones, predicting which ones are most likely to fit snugly into those pockets and interact well.
So computational chemistry helps find the best key for the lock revealed by cryo-EM.
That's a very good analogy, yes. These computational predictions then guide the next step, which is rigorous experimental testing in the lab. You check if the predicted molecules actually bind and if they have the biological effect you're looking for.
And does cryo-EM play a role later on too?
Oh, yes. Once you have promising lead compounds molecules that show potential, you can use cryo-EM again. This time, you solve the structure of the target protein with the lead compound actually bound to it.
Ah, so you see exactly how it's binding.
Precisely. You get a crystal clear picture of the interaction, and that information is gold dust for refining the molecule further, making it bind tighter, making it more selective, reducing potential side effects. It guides the optimization process.
You mentioned an example earlier, PRMT5 inhibitors for cancer. How did cryo-EM help there? Just remind us briefly.
Yes, and just to be clear, that specific paper we noted was in Communications Biology, not Nature, but It illustrates the principle really well from Li Chenglong's research profile. They used Cryo-EM to get the detailed 3D structure of PRMT5, which is important in several cancers. That structure then allowed them to use computational methods to predict how various potential drugs might bind. By using the high-res Cryo-EM structure to carefully evaluate and select the best predicted binding modes, they could then design more potent inhibitors. It shows how powerful combining Cryo-EM with computation can be for cancer therapeutics.
So it's this kind of iterative cycle.
Yeah.
Get the structure with Cryo-EM, use computers to find leads, see how they bind with more Cryo-EM, then use that to make the drug better. Sounds much more targeted.
It's a very powerful and, efficient approach. And it's worth remembering Cryo-EM doesn't exist in a vacuum. It's often combined with other techniques, NMR spectroscopy, sometimes still X-ray crystallography, computer simulations like molecular dynamics.
Putting all the pieces together.
Exactly. Each technique offers slightly different insights. Integrating them gives the most comprehensive picture for making informed decisions in drug design. Cryo-EM has really become indispensable for this rational approach.
It really is amazing how far things have come. So finally, let's look ahead. What are some of the emerging trends? Where is Cryo-EM going next in pharmacology?
Well, the field itself is still evolving incredibly quickly.
Yeah.
The technology underpinning it, the detectors, the image processing software, automation, it's all constantly improving. Meaning we're getting even higher resolution structures, more detail, and we can study more samples faster.
Yeah.
Higher throughput.
So clearer pictures, faster. What else is exciting?
One area that's generating a lot of buzz is time-resolved Cryo-EM. The idea here is to capture molecules in motion.
Like a movie instead of a photo.
Kind of. Capturing those really brief dynamic changes that happen when a drug binds or unbinds, or when a protein just does its normal job. Taking snapshots at incredibly short time intervals. This could give us amazing insights into the kinetics, the speed and sequence of drug action. The actual mechanism, step by step.
Wow, that sounds like it would add a whole new dimension to understanding how these things work.
It really would.
Yeah.
We're also seeing Cryo-EM be applied more and more to bigger, more complex systems. Not just single proteins, but large assemblies of proteins. Or looking at how multiple proteins interact. These interactions are often crucial in disease.
Any examples?
For instance, people have used Cryo-EM to look at how a protein called galectin-3 interacts with another protein, TLR4. That interaction seems to be implicated in Alzheimer's disease. Understanding these complex multi-protein interactions opens up totally new targets for therapies.
So moving beyond single targets to the whole network they operate in, are there still challenges, things Cryo-EM struggles with?
Oh, sure. Studying really small proteins is still quite challenging for the technique. And highly flexible regions within proteins can be difficult to resolve. Clearly, they appear a bit blurry in the final structure sometimes.
Right.
But, the field is actively working on new methods, new approaches to tackle these limitations.
Yeah.
I'm very optimistic we'll see continued progress.
It definitely sounds like Cryo-EM is set to keep pushing the frontiers of molecular understanding for a long time.
I think so, yes. The sheer volume of high-impact research, just looking at Nature from 2020 to 2025, really underscores how transformative Cryo-EM has become for structural pharmacology.
Yeah.
It's giving us these unprecedented insights into ion channels, GPCRs, helping us understand drug resistance, and really driving structure-based drug design forward.
Yeah.
This ability to see things at the molecular level is, without doubt, accelerating the development of new and better medicines.
It really does make you stop and think, doesn't it? Consider the implications of being able to actually visualize the fundamental machinery of life in such detail. How is this ability to see reshaping our whole approach to fighting disease? And, what other breakthroughs might this incredible visualization power unlock in the future? It's been a really fascinating deep dive.